Hello, today I'm going to talk about two useful cellular biology methods, ChIP and CLIP. Chromatic immunoprecipitation, ChIP, and cross-linking immunoprecipitation, CLIP, are methods to investigate interactions between proteins and the genome. Also, they allow to determine which proteins are associated with a specific loci of the genome. ChIP and CLIP workflow could be divided in a few steps. So first, fixation and chromatin cross-linking, followed by cell lysis and DNA fragmentation, then immunoprecipitation, followed by reverse cross-linking and DNA purification, and then DNA analysis. For CLIP, the same steps are applied, but also there are the 3' and then 5' end adapter proteins introduction, and also cDNA synthesis. Just to remember DNA and RNA structures, DNA is a double-strand, negatively charged, entangled with histones, which are proteins positively charged. This structure is called chromatin. RNA is a simple strand, negatively charged. There are many proteins interacting and binding DNA and RNA inside the nucleus, generating specific signaling. So these are the purposes of CLIP and ChIP. How does ChIP work? So here we have a protein A as a target bound to the DNA. So first, we need to fixate protein A in the DNA. To do that, it's added formaldehyde, which is a fixation agent, which will create a specific cross-linking bonds between DNA and the proteins. Next, cells are lysed by sonication, and then restriction enzymes are introduced to cut the DNA in specific sites of interest, generating small DNA fragments. After that, specific antibodies, anti-protein A, coupled with magnetic beads, are added to the solution to bind protein A only. Using a magnetic, protein A attached DNA fragment coupled with the specific antibody is precipitated in the tube. After that, samples are washed to remove all undesired fragments and proteins. Next, it's necessary to reverse cross-linking made by formaldehyde and purificates the DNA. To do that, it's used extensive heat incubation to break formaldehyde cross-linking bonds, and then use specific enzymes to digest the protein component, leaving the protein A attached DNA fragment purified and its sequence ready to be analyzed. Let's talk about CLIP. Here we have now a protein B as a target, for instance. So the same as happens in CLIP, protein B is needed to be fixated in the RNA. To do that now, it's used UV light, which will create covalent and cross-linking bonds between proteins and the RNA. Next, the cells are lysed, and the RNA is cut in specific regions by digestion enzymes such as DNases and RNases, leaving small RNA fragments. Then, specific antibodies, anti-protein B, coupled with magnetic beads are added to the solution, which will bind protein B only. Using again a magnetic, protein B attached RNA fragment, coupled with the specific antibody, is precipitated in the tube. Then samples are washed to remove undesired fragments and proteins. Now it's a new step. So a 3' end adapter protein is introduced in the protein B attached RNA fragment. Then it's necessary to reverse the cross-linking and covalent bonds made by the UV light and purifies the RNA. To do that now, it's used proteinase K, which will digest the protein component and break the covalent bonds. After that, and after the RNA be purified, a 5' end adapter protein is added to the protein B attached RNA fragment. Then a primer is introduced in the 3' end of the RNA fragment, together with the reverse transcriptase to create the new protein B attached RNA fragment complementary strand. And finally, the new protein B attached cDNA is ready, and its sequence is ready to be analyzed. To analyze DNA and RNA, a few methods could be used. Most recently, sequencing for qualitative purposes and qPCR for semi-quantitative are usually the gold standard options, generating high resolution but also high costs. Also, CDS page, Western blotting, and other electrophoresis methods could be applied to analyze ChIP and CLIP samples, giving less quality resolution, but being cheaper options. So when use ChIP and CLIP? ChIP targets the DNA, while CLIP targets the RNA, but it also could be applied for protein-protein interactions. And why use ChIP and CLIP? Because they allow in vivo analysis of DNA, RNA binding proteins. Also, they present high resolution. They also allow the analysis of multiple proteins interactions at the same time, especially DNA histones. And they are standard methods to analyze transcriptional factor signaling and epigenomic interactions. But as all methods, they have a few limitations. So it's difficult in ChIP and CLIP to determine DNA and RNA protein interaction for each cell lysed in the samples. Also, they are dependent on high amount of DNA and RNA in the samples. Antibodies may not efficiently immunoprecipitate all the antigen and protein inside the samples. Also, they have high costs, especially if you use sequencing and qPCR, and they are not suitable for DNA and RNA quantification. Here we have a ChIP application. So this paper describes a protein called NRF2, which is associated with antioxidant pathways, but is also associated with non-small cell lung cancer progression. To find out NRF2 binding sites, the authors performed ChIP on a human lung cancer cell line, which is knocked down for NRF2 using Illumina sequencer. The ChIP-seq results reveal binding sites as genes associated with novel pathways of cell adhesion, migration, and evasion. For CLIP application, we have this paper here, which brings the superoxide dismutase 1, which is an enzyme having antioxidant properties. Mutations in SOD1 could lead to RNA splicing, a genetic cause of a disease called amyotrophic lateral sclerosis. Some studies hypothesize that RNA splicing could be due to SOD1 binding to RNA. To confirm that, authors performed CLIP assay using spinal cord cells from SOD1 mutant mice, then analyzed samples by Western blotting. The UV light and polyclonal antibodies were used in this experiment. However, the results revealed that there is no evidence of mutant SOD1 binding with the RNA. Those are the references that I used, and thank you for watching.